Hi everyone, thank you for joining us today. My name is Jason Kilgore. I'm a technical application scientist in the tech support department here at Molecular Probes, which is part of Invitrogen and Life Technologies. Today's presentation I'm going to be talking about a basic introduction to immunofluorescence staining of cultured cells and tissues. First, a quick introduction. I've been with Molecular Probes now for 12.5 years, 8.5 of that in R&D, making some of these products that I'm talking about today, and doing microscopy for the most part. And for the last 4.5 years or so, I've been with tech support, helping customers like yourself. And I live here in Eugene, Oregon, where we're based. I should also give some credit before we start to Ed Lieber, who's also a tech support scientist here now for 10 years, I believe. He also helped develop the PowerPoint deck that we'll be showing today. So today, we're going to cover a number of topics. These are introductory topics to immunofluorescent imaging. And the presentation will be in two broad groups. At first, I'll discuss the steps of the staining protocol, the materials you'll need, the variations on the protocol, and some of the controls that you'll need. And then I'll go on to a troubleshooting section. There's a decision tree and some of the pitfalls that you might fall into. And by the way, let me say a quick comment about questions. We have a lot of people online today. I think it was 855 last I checked. So there may be a lot of questions coming in. I'll try to answer them as we go verbally. But if I don't get to you, I'll try to answer them at the end of the presentation. And if we run out of time, I'll email you an answer. All of your questions will be saved, and I'll get to them later if I'm not able during the presentation. Here are the very basic sort of steps in the protocol that you'll be following. Observing, fixing, permeabilizing, doing a blocking, including perhaps with a signal enhancer solution, which I'll talk about. Incubating with the primary and then secondary antibody. Counterstaining if needed, and then mounting your sample on your slide. Some of the materials that you'll need include, of course, the cover slips with your cells on it, or your tissue section on your slide. Formaldehyde or other fixative that you'll be using, as well as the buffers to put them in, such as phosphate buffered saline or tris buffered saline. A permeabilizing reagent, such as Triton X100 or Tween 20, for instance. The signal enhancer that I'll talk about. Blocking solutions, such as BSA, or that's bovine serum albumin, or normal goat serum or other serum. Counterstains, such as the DAPI. DAPI is a nucleic acid stain that's commonly used. And then an Antifade mounting medium, such as our Prolonged Gold or Slow Fade Gold. And I'll talk about each of these separately. The first thing that you want to do is look into the cells themselves, right? Before you do any fixation, you should first take a look at your cells in the cell culture area. I'm getting a couple of comments about my volume. Can everyone hear me okay? Please comment if you cannot hear me. Okay, I'll go ahead and continue now. First, you want to observe the cells. So in the cell culture hood, take your cells and take a look at them under the microscope that's there, light microscope. You usually want a confluency for most experiments between 50 to 75%, similar to what you see on the image there. You want them fairly evenly distributed across the cover slip, not areas that would be devoid of cells, and the expected morphology. Of course, cellular morphology differs from cell line to cell line, so you need to be aware already of what's expected for your cells. For non-adherent cells, you'll want to take a look at those as well. Now, morphology is a little harder because they're usually just rounded up balls. But these staining steps can be done that I'm talking about in this in the protocol that I'm giving. You just have to do washes and centrifugations in between each step, or adhere them to a substrate, such as using polyl lysine or a cytospin device. Next, we want to look at fixation. So you take your cells, you remove the media, and you put onto it a fixative of choice. Now, probably the most common fixative out there is formaldehyde. Now, I want to draw a distinction between formaldehyde, paraformaldehyde, and formalin. Formalin has a methanol stabilization in it, so it has methanol contained in it. And for some experiments, that small amount of methanol can actually make a difference. More commonly, people will make their own paraformaldehyde solution from powder, but it's a process that takes a little bit of time, and there's heat, and it's somewhat toxic. Here at Molecular Probes, we tend to buy the electron microscopy grade 16% formaldehyde from various distributors. That solution is kept in small ampules and does not have methanol in it. Formaldehyde cross-links the amine groups on proteins, namely the lysine residues, and that cross-linking between proteins helps retain structure, and thus the morphology of your cells and tissues is better retained. Glutaraldehyde fixes more strongly. It has twice the reactivity. The problem, however, with aldehydes is that they can increase autofluorescence. Autofluorescence is the native fluorescence of your tissues or cells. It's particularly a problem in like paraffin tissue. Paraffin processing and fixation can cause a greater degree of the background autofluorescence. And fixation with glutaraldehyde actually increases that beyond what you would see with formaldehyde. So for most fluorescent microscopy usage, you want to avoid glutaraldehyde for the autofluorescence reason. Now, there are some exceptions. For instance, when you're fixing endoplasmic reticulum, sometimes you need a stronger fixation. Now, to answer one of the questions that came in, yes, you can, of course, stain cells without fixing, that is, staining live cells. Now, those antibody labeling techniques typically are for the cell surface, such as for CD antibody labeling in flow cytometry. But for most microscopy usages, at least, you're usually looking for intracellular, and when you do that, you need to fix and permeabilize. Now, other fixatives include methanol, which I misspelled there, sorry, and acetone. Now, let's go to the next slide here. I think I addressed those. Well, I didn't address it there, but methanol and acetone fix in a different way. They precipitate proteins. But they also delipidize your sample, which means you can lose some proteins. And because you're not doing a cross-linking, sometimes cellular morphology is negatively affected. You can get shrinking of tissues, for instance, or cells for that matter. Some antibodies, some primary antibodies require one fixation or the other, or rather they have been validated for one fixation technique or the other. So you'll want to check that when you purchase your antibody and see what technique they had used. Now, for fixing using formaldehyde, you always want to use fresh formaldehyde. I've kept it in the fridge at 4 degrees for up to a day or two without any apparent drawback, but it does lose its fixative ability. So generally you want to make it fresh. And typically you'll make a stock solution, maybe 16 to 30%, and dilute that down into PBS at 2 to 4%. And then you fix your sample by immersing them in the formaldehyde. Usually 15 minutes is very typical, sometimes longer. Tissue samples often will be kept in formaldehyde solutions at 4 degrees for several days. But for cell fixation, usually 37 degrees for 15 minutes is sufficient. You can't really prepare your formaldehyde and freeze it to use later on. That would likely lose its reactivity even despite the freezing. Most fixation methods require the formaldehyde fixation, to answer one of the questions. And as far as what you do the fixative in, some people will use PBS. I generally recommend using complete media because it's better for the cells. Some cells are so sensitive that in the few minutes before they fix, just putting them in a cold solution or in PBS instead of media can cause cell morphology problems and even toxicity in that short time. So generally I recommend making it up in media. Okay, going on. Now once you've fixed your cells, you'll give it a couple of washes in PBS or other physiological buffer. At this point, you no longer need to keep it at 37 degrees for most usage. You can do things at room temperature since your cells aren't alive. Using a detergent actually sort of pokes holes in the cell membrane, if you will. It delipidizes the membrane, allowing entry of large molecules like antibodies into the cytoplasm and nuclear compartment. Triton X100 is a very typical detergent that's used. Anywhere between 0.05 and actually 1% I've seen as high as. Most typical would be about 0.2% in PBS. Sometimes you'll see in protocols the mention of PBST, which is phosphate buffered saline with Triton X100. A typical time for the permeabilization with a detergent could be as short as 5 minutes. I typically would go 10 to 15 minutes. Sometimes people will do an acetone permeabilization. Acetone delipidizes quite strongly. This can be a problem for some proteins, especially membrane proteins. Doing the acetone protocol, you would use it pre-chilled at minus 20 degrees and exposed to the acetone only for about 10 minutes. Some people will use a 50-50 methanol acetone for fixation and sometimes for permeabilization. If you fixed your cells using a solvent, like methanol or acetone, you usually don't need to follow up with the permeabilization since it's already delipidized and now large molecules can enter into the cells. Once you've done your permeabilization, you can wash away any of the perming reagent with PBS and then go on to your blocking steps. Now, when I say blocking, typically when someone says blocking, they're talking about a protein blocker, which I'll get to in a minute. But there are other blocking steps that you can do. Just very briefly, I'll mention, for instance, if you're doing streptavidin biotin labeling, you'll block for endogenous biotin in your cells. If you're doing an HRP or horseradish peroxidase labeled secondary, you'll block for endogenous peroxidase using hydrogen peroxide. Here, I mentioned the use of the Imagit FX signal enhancer, which is another blocking solution. This one blocks a different sort of thing that most people don't tend to think about. Dyes have a certain charge to them, most of them. Most dyes have a negative charge. For instance, the Lexafluor dyes are negatively charged. And when you label your cells or tissue, that negatively charged dye can be attracted to positively charged areas in the cells or tissue. In cultured cells, those positive areas are the nuclei and mitochondria mainly. In tissues, it can vary. For instance, in brain cryosections, the myelin or white matter is positively charged, and the dye goes to it, and you wind up with nonspecific labeling of the white matter. This signal enhancer solution actually blocks those positively charged areas and thus reduces the nonspecific dye binding. I helped develop this product, and I got to where I used it every time I did a label. Not all dyes are as bad as others. Here, for instance, you see tetramethylrhodamine, which is a common orange fluorescent dye, and how it's binding to the nucleus and mitochondria in some cells on the right. On the left, we have some blocked cells. After you've done your ImageFX signal enhancer, then you can go on to your protein blocking. Now, there's a lot of different blocking reagents out there. Some are commercial. Very commonly, people will use serum from the species in which their secondary is raised. For instance, if you have a goat secondary antibody, you'll use normal goat serum. I recommend between 5 and 10% normal goat serum or other serum. That's very common. Another common blocking reagent is bovine serum albumin, or BSA. 3 to 5% is commonly used. When I would do a protocol, I would typically do a mix of those two, a 6% BSA with a 10% normal goat serum or other serum. And I found that that was very good for blocking. Sometimes people will use other things like casein, which I have found not to be very efficient at blocking nonspecific background. Same goes for fish skin gelatin. Sometimes people will have good use with that, but generally BSA and serum is the best option. Now, we also carry Blockade blocking solution. That's our commercial blocking solution, which is a mixture of various protein blocker. We actually market it for blocking microspheres, but if you use it undiluted or only lightly diluted, it's as good or better than anything else out there, I've found. Typically, you'll block your sample for cultured cells, typically 30 minutes to an hour. And you'll make your primary antibody in that blocking solution. Now, for tissues, as with all steps with tissues, you'll go longer, often blocking for an hour or two. I skipped a slide there. Next, you want to incubate with your primary antibody. But of course, when you're purchasing your antibody, there are some important things to think about, right? First, of course, you want it to be against the target of interest. Not all targets are commercially available, so you might have to have one custom made. There are some good sources for commercial primary antibodies. We sell quite a few here at Life Technologies. There are a number of other companies out there, such as Santa Cruz or Southern Biotech. There's also some online sites where you can compare and search through a number of companies. One is biocompare.com. The other is linscottsdirectory.com. That's, if I remember correctly, LINNSCOTTS directory.com. Those are very good sources. Once you've found an antibody against your target, you want to make sure it's been validated to use with your species of cell or tissue and has been validated for your method of imaging, like for microscopy or for immunocytochemistry or immunohistochemistry. Some antibodies have only been validated, for instance, for Western blot. And I can tell you from experience that those that are validated for Western blot don't always work for immunocytochemistry. So you need to be careful and check the information on that, which is usually on the manual for the antibody of interest. The antibody concentration to use can vary. Typical concentrations can be anywhere from 0.5 to 10 micrograms per mil. Companies don't always tell you the antibody concentration. Often they'll give just a dilution factor or a range of dilution factors. So you want to check that. Now, I always recommend when you first get the antibody to do a range of concentrations or dilution factors to find out what's optimal on some control cells first. Incubation time for the primary antibody, typically 30 minutes to an hour for cultured cells. For tissue, it often goes much longer. It can be several hours, 2 to 4 hours at room temperature, sometimes overnight at 4 degrees. Once you've finished your primary antibody, you'll want to wash it very well to remove any primary. If you're using tissue, you want Triton or another detergent in all washes, as well as in with the primary and secondary. Now, your secondary antibody, there's a number of things to consider when you're purchasing your antibody. First off, you can see there are a number of classes or types of antibodies. Whole IgG is the most common. This gives the strongest signal and usually is the least expensive option. A Fab fragment or Fab2 fragment is another option. They're smaller. They're able to penetrate a little bit better, and they don't interact with Fc receptors. They can be more expensive, and you get lower signal due to fewer dye molecules per antibody. If you're doing multiple antibodies on the same sample, you want to make sure that they're of different species. So your primary antibodies, you usually want different species, like one mouse and one rabbit, so that they don't cross-label. And you want them to be highly cross-absorbed. That is, they're cross-absorbed against the other species. So your mouse antibody, if it's not cross-absorbed against rabbit, it might label your rabbit primary, and vice versa. We sell a number that are highly cross-absorbed, or you can check the manual of your secondary antibody to see if it lists what species it's been cross-absorbed against. And those tend to be a little more expensive too. And also you can have secondary antibodies against specific isotypes like IgG1 primaries or 2A or 2B. If you have two primary antibodies of the same species, like two mouse primaries, if you're in luck and you wanted to use them both on the same sample, one might be an IgG1, the other might be an IgG2A, and there are isotype-specific secondaries that target only one or the other, and thus you can use them together on the same sample. If they're both mouse IgG1, then your anti-mouse secondary would probably label both, and you don't want that. The final consideration in choosing a secondary antibody is the wavelength, right? Choosing the fluorophore or dye that's on your secondary. Very important. First, you want to look at your microscope or confocal microscope and find out which wavelengths or filters are available, and try to match your dyes to those. Very common wavelengths might be the DAPI wavelength or Alexa Fluor 350. That's your typical blue wavelength for a DAPI filter. Fluorescein or Alexa Fluor 488 for green. Orange would be like Alexa 555 or tetramethylrhodamine. Red could be in the Tritzy wavelength or rhodamine red or Alexa 568, or Texas red or Alexa 594. And more commonly these days you'll see a far red option, which is beyond our visible wavelength, but cameras can see it quite well. For instance, the Psi-5 or Alexa Fluor 647 wavelength. If you're using a laser-based system like typical for confocal, you can use Alexa Fluor 633 or 647 excitation with Alexafluor 633 or 647. By the way, I got a request here to state the resources for antibodies again. Let me just repeat. One is biocompare.com. The other is linscottsdirectory.com. That's LINNSCOTTS directory, linscottsdirectory.com. Okay. Once you have your secondary antibody, and here you can see in this next slide the various different Alexafluor color options that you have. Once you have your secondary antibody, you'll want to have an incubation time. Before I get to that slide, let me just say that Alexafluor dyes, you saw the various different colors we offer. Our Alexafluor dyes tend to be more stable and photostable than other dyes. And I'll talk about photostability and antifades in a minute. Here we compare Alexafluor 488 to fluorescein. For instance, after only a 1-minute exposure, the fluorescein dye has pretty much gone away, whereas the Alexafluor 488 is still fairly strong. You can extend the photostability with antifades, which I'll talk about later. Our Alexafluor dyes are also very pH insensitive, so at low pH they're not going to dim as much as, for instance, fluorescein. And we have common dyes for the common filter sets. Now, when you're incubating with a secondary antibody, note that a typical concentration can range between 1 and 10 micrograms per mil. Most of our secondaries here at Molecular Probes are 2 mg per mil. So you'll dilute maybe 1 to 400 would be very typical. That would give a 5 microgram per mil final. You want to briefly centrifuge your secondaries as well as your primaries prior to using them. Centrifuge the stock solution. Sometimes there can be solution in the cap and there can be aggregated material in solution. You want to spin that down. If you don't spin down, you can wind up sometimes with spots in the background, which is aggregated antibody. And you'll dilute your antibody into a like 6% BSA in PBS. It's very typical. And incubate for 30 to 60 minutes for cultured cells. Again, with tissue, you often go longer, sometimes a couple hours or even longer to get good penetrance. And I recommend for both primary and secondary antibody labeling steps and counterstains too, to do the incubation in a humidified chamber of some sort so that you don't get evaporation effects concentrating your dye more than you want it to be. Once you've done your secondary antibody labeling, then you give it a wash and do any counterstaining that you might want to do. Counterstains, most typically people will do a nucleic acid stain such as DAPI, which is a typical blue fluorescent nucleic acid stain. It's very highly selective for double-stranded DNA. And most microscopes have a filter set for DAPI. If you're using confocal, you'd use a UV laser to excite it. Other common counterstains might be, for instance, phalloidin conjugates, which will label the F-actin in cells. Here you can see various colors of the phalloidins that were used and some images that I took. The F-actin gives a very general outline of the cell cytoplasm because of the abundance of actin filaments, which actin is a cytoskeleton component in cells that help maintain morphology. Other counterstains might be for various organelles like endoplasmic reticulum or Golgi or mitochondria. We've got a lot of different dyes here, but I'm not going to address those other organic dyes for organelles in this particular presentation. Labeling times and concentrations vary. DAPI, for instance, typical time and concentration would be 0.2 micrograms per mil. Label time may be just a couple minutes in PBS. Now, once you've done your your counterstaining, you're ready to mount your sample typically. You'll give it a good wash in PBS. Then you take your cover slip and just lightly touch the tip of it to a chem wipe to wick off any excess buffer. And then you turn your cover slip over onto a drop of your mounting medium on your slide. Now, there are a lot of different commercially available mounting media out there, and some people make their own even. I highly recommend using an Antifade mounting medium. An Antifade mountant helps slow the photo-bleaching of a dye. That is, when you shine light on your dye under a microscope, it starts dimming or photo-bleaching away. This is, for the most part, non-reversible, so your dye goes away and stays away. Using an Antifade mountant, you slow that photo-bleaching considerably for most dyes. Our primary Antifade mounting media here are Prolonged Gold and Slow Fade Gold. Prolonged Gold, the difference between these two, they have basically the same components, but a different base in them. Prolonged Gold, you'll mount your sample and then let it go overnight, and it cures or hardens to a firm gel. That gel allows it to be more archivable, and you can keep your sample at 4 degrees for weeks, potentially. Slow Fade Gold does not firm up. Instead, it stays liquidy, sort of like a glycerin or a glycerol solution. That is, it's non-polymerizing or non-hardening. And that's fine if you want to just take a look at your sample right away and then throw it away and don't care to archive. There are a lot of other mountants out there. You want to check to make sure that they have an Antifade component in them. Shield is another common one that's used, although it has a blue autofluorescence, which can be a problem for blue dyes, and it doesn't protect quite as well dyes in the red or far red as ours. So that's most of the protocol basic information that I had to share. Let me just take a real quick look at some of the questions here before I move on to controls. A long blocking period between antibodies. No, typically you only want a blocking step right at the beginning before you do any antibody staining. You don't need to do separate blocking in between. One person asked if Triton X should be used between blocking primary, secondary incubation steps. Generally, I recommend including the detergent in with all washes and incubations, especially with tissue. Now, with cells, you can get away with just doing the permeabilization at the beginning for the most part. Someone asked what highly cross-adsorbed means. That's where antibodies are basically cross-adsorbed against other species, so they don't label those species. So your goat anti-mouse, if it's cross-adsorbed against rabbit, it's much less likely to label rabbit primaries non-specifically. And yes, to answer another question, you can use blockade for diluting your antibody, that is your primary antibody or even your secondary antibody, and it works great. One last question here before I go on. What speed do you spin your antibody stocks to bring down the aggregate? Basically, I'm talking about one of those benchtop centrifuges. We call them turtles here. You just put it in and bring it up to the top speed and then let it go back down. That's usually sufficient. Okay, moving on here. Let's talk for a minute about controls. There's a number of different controls you want to do when labeling your cells or tissues. First off, I mentioned earlier that cells and tissues can have an autofluorescence. That is, they fluoresce in visible wavelengths. And I think I'll show you a picture of that here in a minute. So it's important to have cells or tissue section with no dye at all, no antibodies and no dye, imaged in the same way and mounted in the same way to just see what that background fluorescence is. Hopefully it won't have any, but typically tissue in particular will, especially for paraffin sections. For a no primary, you also want a no primary control. This is because if you see background in your sample, you don't know if it's coming from your primary antibody or from your secondary. So having a no primary control will check to see if there's background binding just from the secondary. And I'll talk a little bit in the troubleshooting section more about these controls. And I'll have a decision tree also coming up about those and some suggested solutions. Right. Secondary antibody background, there's a number of things that can cause the background. For instance, if you have too much of your primary or secondary antibody, when you start getting too much of a dye or an organic dye or an antibody, you start increasing that non-specific background. And so it's always important to run a range of concentrations, both of your primary and secondary, in some controls before you begin your main samples. And you'll see that with too high of a background, you can start getting that non-specific binding, both on cell and off cell. Also, you want to make sure you're blocking sufficiently. I went over blocking solutions earlier. If you're getting a lot of background, you might want to make it more stringent, that is, going longer or a higher concentration of your blocking components. I mentioned dye sample interactions. That's dye charge binding, negative dyes binding to positive areas. Antibody aggregates, you want a centrifuge to remove those. Typically you'll see spots in the background if there's aggregated material. And some dyes just go bad, or some antibodies just go bad over time. Most of our antibodies have a guarantee period of 3 to 6 months. So you want to check with your distributor, find out what their recommendation is for storage and stability time. If it gets too old, you start losing specificity and increasing the background until finally you get no labeling at all that's specific. If you get no signal at all, then there may be some other issues going on. Either your primary or secondary has just completely gone bad and isn't labeling anymore. You want to make sure that your primary antibody is actually against that target that you're looking for and has been validated with the species of cells that you're trying to label and has been validated with the technique you're using, Western blot, for instance, versus immunocytochemistry. Filters, you want to make sure that your filters are appropriate for your dye. We have an online tool called the Spectra Viewer. You can go there to check. You can enter your filter settings in it and compare it to dyes. So go to invitrogen.com, look under the Resources tab, and there will be a link for the Spectra Viewer. Other filter companies sometimes have some very good tools online. For instance, omegafilters.com. They have what they call the Curvomatic, which is a tool where you can enter their filter specs and compare it to different dyes. Sometimes you get little or no signal just because there's very little of the antigen in your cells that you're targeting for. It may be a low expressing antigen. Poor fixation, if you underfix or even if you overfix, you can get very little penetrance of the dye or of the antibody into your sample. If you've underfixed, or certainly if you've underpermeabilized, the antibody may not be able to get in. If you've overfixed, it may be too highly cross-linked. An antigen retrieval method can help remove some of that cross-linking. And some antibodies actually require an antigen retrieval method. Antigen retrieval, there's a lot of different techniques out there, but basically it involves heating your sample, often in a special solution like a citrate buffer or citrate urea buffer. Some commercially available pressure cooker devices are out there that combine pressure and heat to do an antigen retrieval. There's a lot of different methods. And finally, your secondary antibody, you want to make sure it's against the species of your primary and that it's been used against similar primaries in the past and is of the appropriate concentration to use. Now, I mentioned autofluorescence earlier. That's the endogenous fluorescence of your sample. Plant samples, for instance, have a very high autofluorescence due to, for instance, chlorophyll. Old brain tissue, especially from older mice or humans or whatever age the organism was, can build up lipofusion, which is a protein that autofluoresces. Autofluorescence can come from overfixation, especially with glutaraldehyde, and tissues have much higher autofluorescence. If the autofluorescence is from your fixation technique, you can do a sodium borohydride fixation. Just very briefly, sodium borohydride is a component you can buy from chemical companies like Sigma. You'll make it extremely fresh, so you have powder in tubes, and after you've done your permeabilization, but before you do any blocking, you would wash it 3 times, 5 to 10 minutes in a 1 mg per mil sodium borohydride solution in PBS. You make it extremely fresh, so you put in your PBS, vortex it, and put it on your sample. And in tissue, you'll see it fizzing. And then after 10 minutes, you make up your next tube, very fresh, and put it on, and then you do it a 3rd time. This especially helps in the green and red wavelength. Some cellular compartments are just fluorescent, for instance, NDH or NADH in mitochondria. Autofluoresces in blue. You can block this with compounds like Sudan black, or you can photo bleach it away prior to incubating with your primary. There are papers on these various different techniques. I've even heard of people taking their samples and putting it in strong sunlight to photo bleach away the autofluorescence. You can also keep one of your channels open and use the autofluorescence in that channel and subtract that image from your specific channel image in a different wavelength using the arithmetic functions on your imaging software. Finally, if your dyes are too strong, you can actually get bleed-through from one dye into the other. That's not really autofluorescence. It's what we call bleed-through. And you can reduce your dye concentration. Sometimes your autofluorescence that's endogenous to the sample will be brighter in one channel than another, and so you can just avoid those channels. And here's a general image showing how autofluorescence in a tissue sample is very ubiquitous through all the channels. It's a very broad excitation and emission. Here I show the typical 3 channels, green, blue, and red, and then an overlay of them of a kidney sample, showing how it's throughout the wavelength. Oh, finally, some imaging devices like Zeiss 510 Meta confocals, for instance, or Leica SP5 will have spectral deconvolution systems where you can subtract out the autofluorescence just based upon its spectrum. There are some problems that can arise in background due to secondaries. Too much secondary can cause increased background. You can see on the left where we've had too much. It's actually extremely bright, and here we've saturated the signal in trying to label a tubulin. And on the right hand, we've used less of the secondary, and you can see a much better tubulin labeling without all the background. Now, primary antibodies, looking at the background that you get, if you've done a no primary sample and an autofluorescence control and seen no background, then most likely your background was due to your primary antibody. So you want to make sure it hasn't gone bad over time, that it's been stored properly. If you see a punctate background, it may be due to the need, due to aggregates, so you need to centrifuge. Or you may just need to find a better primary. I've bought some primaries new from some companies that just weren't very good and they weren't very specific. In that case, contact, of course, the tech support of the company that you got it from. I mentioned antigen retrieval earlier. Here's some example images where antigen retrieval, of course, the different methods vary widely. I'm not sure if you can see on your screen, it might not come through, but on the left-hand side, we have a sample where it was labeled with an anti-Golgi antibody. It's labeled the Golgi bodies. And without antigen retrieval, there was a very high punctate background and a lot of on-cell background. Doing an antigen retrieval method cleared that up. Now, we made a little bit of a mistake. We meant to show the same primary in both images here. I think we messed that up. On the right-hand side is a mitochondrial dye or antibody. I think it was one of our anti-OXPHOS antibodies. And antigen retrieval and urea cleaned it up. Prior to doing the antigen retrieval, there was actually very little of the specific signal and a lot of on-cell background. Again, contact your antibody distributor to find out if your antibody needs an antigen retrieval method. Let me just take another quick moment to go over a couple more recent questions, and then I'll give a conclusion. To answer one question here, what kind of antigen retrieval is good for cultured cells without losing them? Antigen retrieval is less often used for cultured cells than it is for tissue sections, but it can still be needed. Most commonly, just a citrate buffer at 95 degrees and a double boiler is good, but again, it varies from antigen to antigen as to which method is best. Basically, you lower your, in the double boiler method I mentioned, you have a little Columbia jar, which is a little multi-cover slip glass jar with your antigen retrieval solution in it that's in a second container that's on a heat block with water. And then when it comes to temperature, you just lower your cover slips into that and let it sit for 30 minutes or so, and then you take it out and put it back into room temperature buffer. Another question here is appropriate speed and time centrifuge the primary. Again, just bring it up to speed, up to full speed with a typical benchtop centrifuge, and then let it come back down and use the supernatant. If you see a really big pellet, then chances are your antibody is no longer stable. But if it's just a little bit at the bottom, then I wouldn't be too concerned. Temperature for storing antibodies, 4 degrees is often fine for short term, but for longer term storage, you'll want to aliquot and store at -20. Some distributors suggest making it up in a 50% glycerol solution to help prevent it from freezing solid, because repeated freeze-thaw can sometimes degrade your secondaries. Okay, one more question here before I do the conclusion, and then I'll answer a few more after the presentation. One person asked, how do I make fresh formaldehyde? Just do a quick search online for paraformaldehyde recipes, and that will you'll find them there. To block endogenous biotin, another person asked about doing that. There's a number of blocking kits that are available commercially, which I recommend using. We have an endogenous biotin blocking kit here. So if you go to invitrogen.com and type in endogenous biotin, the kit will come up. We actually have 2 different kits, I think, which are pretty much comparable. They're just from different parts of the company. And it's a two-step procedure. Takes about 30 minutes or so. There's also kits from other companies. Vector Cell's a very good one. And then finally, for Alexafluor dyes, is it wise to aliquot and store at -20 or 4 degrees? I think I answered that already. 4 degrees for short term, -20 for long term. By long term, I mean more than a couple weeks. For no primary controls, would you still add a secondary antibody? Yes, that's what I'm talking about. So no primary control would have no primary, but you would add the secondary antibody. Okay, let me just go on to the conclusion real quick. It will let me. There we go. All right. So first off, let me just say that not every protocol is the same, right? You have to optimize your experiment. You have to optimize your fixation technique, the concentration of your primary and secondary, and the label time. Maybe try a couple different antigen retrieval methods if needed, or blocking solutions. So before you try a new primary, you want to try some of those controls first, find out what's best. And of course, run those controls with every time you do it for the first time. I say with every experiment, they may be a bit much. Certainly the first time you use a primary or secondary, you want to use the control, or if it's been a long time since you've used it, or if in some other way you've changed your protocol significantly. I also want to say just real quick that you want to optimize your imaging system, your microscope, not just for the correct filter, but make sure everything is aligned, that the illumination source is correctly aligned, that the background is made uniform through the software, and that the condensers are aligned. Talk to your scope facilitator about that, about the hardware issues, and that you're familiar with using your software, of course. Of course, wherever you buy your primary or secondary from, feel free to contact your tech support. Having done tech support myself for 4.5 years, I really love talking to you guys about the protocols and helping troubleshoot. Here on screen, you can see some contact information for us. Again, my name is Jason, but anyone at this phone number or at this email will be happy to help you. We have a team of 12 tech support scientists here. I was in R&D as well. Another one was actually a manager over in R&D for 10 years doing conjugates. All of us have been in the lab developing products at different companies, at least, if not here, and are very well, most of us have PhDs, so, we're good. You can give us a call anytime or write us. And if I'm not able to answer your question here in a minute, I will send you an email with your answer. I have your emails here as well as your questions. But if I don't get back to you in a timely fashion, just feel free to call or write and we'll get back to you. Okay? Well, thank you very much for attending today. I believe this will be made available online. Yes, it may take a week, but I'm hearing that will be available online at some point. If you wanted to listen in again and go over it again or share it with others. And I'd like to thank the administrators who helped put this together. This website will be or this presentation will be on the seminars page at invitrogen.com. Okay. Well, thank you very much again, and I'll go ahead and answer a few more questions that people had sent in. Okay, so going back through some of the earlier ones here. Evans Blue for counterstains. If I'm correct, Evans Blue is a chromogenic dye. That is, it's a non-fluorescent dye. I didn't address chromogenic dyes that you would use with transmitted light to image. Most often, chromogenic dyes will actually quench fluorescent dyes, so you don't typically want to use chromogenic and fluorescent dyes together. Now, there are some exceptions, but for the most part, that remains true. Is it possible to incubate with different antibodies simultaneously, or should it be done separately? Well, as long as your primary antibodies are of different species, or at least of different isotypes, then you can do them together. So you mix them together in one solution and put them on together, wash, and then mix together your secondaries, as long as they're cross-absorbed against each other, and in one solution and wash. Now, there are occasions where you want to do a sequential labeling, but it's not often recommended. Usually people will do that because of concerns about cross-labeling between species, but it's not always very successful. Now, someone, I forget where it was in the list here, but someone had asked about doing labeling with direct labeled secondary or primary antibodies. That's one example where you might want to do a sequential labeling if it's of the same species. So you'll have your unconjugated mouse primary, for instance. You'll label that first, then your goat anti-mouse secondary, wash, and then you can come in with your other mouse primary, which is already labeled directly with another dye. And so that's one instance where you can do it. Now, going on more about that question about the direct conjugates are dimmer and a little harder to penetrate into tissues, certainly, but sometimes cells too. And so direct conjugates, often it's not used if it's a dim or a low-expressing antigen. For tissue, the autofluorescence is often strong enough to overwhelm your dimly labeled primary that was direct conjugate. It can be done, and I've done it, but those are some concerns, mainly about the dimness of the direct conjugates. Now, direct conjugates are very often used in flow cytometry as opposed to microscopy. In flow cytometers are more sensitive, so the dimness is not as much of an issue. Plus, a great deal of flow cytometry targets are cell surface antigens, so penetrance is not as much of an issue. Okay, are Antifade agents available for live cell staining? Not really. That's something that I've struggled with here as well. In live cells are much more sensitive to different components in terms of toxicity. The Antifades that you can buy commercially are for fixed cells and tissues, and those will kill your live cells. And I've had very little luck trying to develop a live cell antibody here, so I won't go into detail on that. There are some homebrew recipes available online. Most commonly, people will use Trolox, which is a water-soluble vitamin C, I think it is, if I remember correctly. Sometimes people will use other vitamins to block, like vitamin C, for instance, or ascorbic acid. I've had very little luck with those. For the most part, they don't protect the dyes very well. For live cells, really, the best option is to either fix them or to try to just limit the light exposure. Sometimes if your dye is really sensitive and photobleaching quickly, it can do to have a second dye that's more stable, like Herxt 33342, which is a blue fluorescent live cell nucleic acid stain. You'll use that dye to find your cells and get into your focal plane, and then you'll switch over to your photobleaching dye and take a picture real quick, right? Can Antifade reagents work for any dye and any wavelength? Not really, actually. they'll work to some extent, but some dyes work better with different antifades than others. Now, our antifade mountants, Prolonged Gold and Slow Fade Gold, work for most any dye, but there are a couple that it's not as good for. For instance, our Alexafluor 532. Not much works for that dye, but generally it'll work for pretty much any of them. Like I said, Vecta Shield, for instance, in the far red won't work so well. Be careful, some antifade mountants that are advertised as antifades actually don't have components that are antioxidants that would stop the photobleaching. Instead, they just cure hard, which can slow photobleaching. But that's not always sufficient. So you need to check that with the company that sells it. How do antifades inhibit photobleaching is another question. Last question. Basically, they have the good ones, like ours, have an antioxidant component or free radical scavengers. When a dye is exposed to light, it creates free radicals and singlet oxygen, which can damage neighboring dyes and cause photobleaching, and its source spreads. If your mountant cures or hardens, it can slow the spread of those reactive components. And if you have a free radical scavenger or antioxidant in there, it will mop up those free radicals or singlet oxygen and prevent it from hurting other dyes. One more here. How does Fluoramount G from Southern Biotech compare with Prolonged Gold? As far as I can tell, Fluoramount G, although it hardens comparable to Prolonged Gold, I'm not aware of it actually having any free radical scavengers in it. So that's one of those that its main antifade component is just that it cures hard. My comparisons have shown Prolonged Gold to be somewhat better for most dyes, if not all dyes. And, I can't help myself. One more question. Why use warm fixative? Some cells are just so sensitive that they can change morphology and have toxicity prior to the effective action of the formaldehyde. Okay, well, I think that's all the questions we have time for today. The rest of you I'll try to answer offline in the following couple days. Thank you again so much for joining us today, and I look forward to hearing from you in the future. All right, bye-bye.